Okay, good morning. Today, let's take a closer look at issues that concern the uncontrolled proliferation of cells. We've been discussing how important it is to consider that cell proliferation and movement through the cell cycle is a constant sort of tug of war between our oncogenic signaling pathways in the cell that respond to external signals that are arriving from the outside, being counteracted with the different sets of diverse breaks that we have inside of the cell in the form of tumor suppressor genes. And it's that back and forth, that sort of two-footed driving of the cell, as I like to describe it, that ultimately determines which way the cell cycle progresses. Does the cell favor differentiation and stay in G1, or does the cell move forward past the restriction point and head into mitosis? Right? So let's take a closer look at that. Let's review some key concepts here. So we said that normal cells listen to environmental signals that govern their proliferation. Right? And we said that importantly, this happens during G1. The cell cycle can halt and resume before completing mitosis if along the way the cell encounters certain problems, for example, during DNA replication, right? Or if there are sudden changes in the cellular environment, like depletion of certain vital nutrients, that may jeopardize the ability of the cell to finish going through mitosis. And of course, a key concept that we've always been having in mind throughout the semester is that one of the hallmarks of cancer is that cancer cells have the ability to divide many more times than a normal cell would. So this, I want you to think about this. When we talk about a cell that is moving out of G1 into S phase, and the cell begins to undergo the events of duplicating the genome, right, replicating all of the chromosomes during S phase, and ultimately after it goes into mitosis, right, those replicated chromosomes, the sister chromatids, are going to divide to form two daughter cells. Right? When we think about that genetic material that's being copied and passed on, are the proliferating cells really identical copies of each other? Or is it more like we are dealing with photocopies of photocopies that lose information with every round? I think most of us don't think about that, but then when you actually do stop and think about it, you realize, yeah, in some ways, right, what we're doing is just repeatedly making copies based on pre-existing copies. So if you have, for example, a photograph of a friend of yours, right, and you make a photocopy of that, let's say you wanted to make, like 50 copies of this photograph. Right? So if you put it in the photocopy machine, all of you just put the one original copy in the photocopy machine, hit copy, and it makes 50 copies that are all based upon that original image, right? But what if another way you could do it was you just make one copy of that, right? And then you say, well, I need another copy of it. So you use the first photocopy to make the second, and so on and so on and so forth. With each successive round of photocopying the pre-existing photocopy, you're going to lose some information, right? So in the case of your photo, maybe by the, I don't know, fifth photocopy of a photocopy, you begin to no longer have enough information there for you to really accurately identify, oh yeah, this is my friend, right? So is our DNA, is our genome any different? I think all of you, now that you are all upper division students in biology classes studying cell biology, you already have an appreciation, right, for how much information is in our genes and in our genome. And a lot of that information is not even the actual genetic sequences of coding regions of genes. A lot of that information are the subtle types of bits and pieces of information surrounding genes, right? Things like what kinds of sequences are present in promoters or other type of regulatory elements that ultimately, right, finely tune how different genes are expressed. Adds a lot of very, very subtle, nuanced, detailed information. Right? And yet, every time you copy that information and make daughter cells that then divide to make additional daughter cells, right? Aren't you, in many ways, doing this type of thing, making photocopies of photocopies, right? And isn't it possible that you lose information along the way? Right? So here's a thought question. Every cell in our body today, as you're sitting there, and we're having class together, right? Every cell that makes up your body can trace its origin back to that single fertilized egg, right? 20-something years ago, right? What would the sort of family tree of cell generations look like if we were to actually follow that original single cell becoming 10 to the 14th cells over 20 plus years? Right? Let's try to imagine this, because I think it's an interesting kind of thought question. Would it look like how we typically imagine something like a family tree? So here is, I think what we all sort of think of when we think of a family tree, like if you do your ancestry.com or whatever, right? So you have generations that of great grandparents, right, giving rise to grandparents, giving rise to dads and moms, that then eventually give rise to you, right? And then here you are with your siblings, right? So this is how we usually picture a family tree. But if we were to imagine the family tree of cell generations going from one cell to many, many cells, we would say, nah, it wouldn't really look like this, right? It would be kind of like the opposite. You're sort of going from bottom to top. Right? So it would look something more like this, because each generation would represent two daughter cells coming from that previous cell. So you might say that if you were to imagine over time this family tree of one fertilized egg becoming you, becoming the 30-something trillion cells that you are today as you're sitting here in lecture, we can imagine that there are early generation cells, mid-generation cells, and late generation cells. Towards the bottom, right, you have many, many cells that have come from the original starting one. But what would happen if our cells kept dividing on and on and on like this, right? So this last generation down here, you see each little fork showing a division. They don't even bother showing a cell anymore, right? Because it would just get too many of these, but each one of these little things would be a cell. And then what if that just kept dividing and dividing and dividing? Well, we would be a big mess, quite literally, right? We would not have our normal sort of human body shaped the way that we're shaped. We wouldn't be able to function the way we are, because we would just be one humongous, massive tumor, right? That's disgusting, right? But hey, but it's true though. So think about how is it that all of these trillions and trillions of cells, these fully differentiated cells that you see when you look at yourself in the mirror today, right? How do they actually know when to stop? So there must be lineages, or sorry, there must be limits on different cell lineages. So instead of calling these early cells, mid cells, and late cells, let's get a little bit more detailed here. So in the early embryo, of course, starts from the original fertilized egg. That fertilized egg sort of would be the ultimate pluripotent cell, to the point where we actually call it the totipotent cell, right? The one that starts it all. And then that fertilized egg begins to divide, and you start developing the embryo. Then along the way, you have many pluripotent cells. Right? So these are still cells when the embryo is maybe just like a volleyball made up of 32 cells, 64 cells, right? Each one of these cells is still capable of becoming any cell eventually in the body. Right? Then they move on to differentiate further and become multipotent stem cells. And importantly, these multipotent stem cells then divide and fully differentiate to create the trillions upon trillions of fully differentiated cells in your body. So if this, in one sort of simplified cartoon, is the history of your life, right? Where are you today? What are the cells in your body today? Like, where would they be on here? Well, they'd be here. Right? So the cells in our body today, we no longer have pluripotent cells, right? All of our tissues are populated with multipotent stem cells that persist indefinitely and continuously give rise, right, to daughter cells that continue differentiating and become, in each case, maybe one of a handful of different specific fully differentiated cells that multipotent cell is tasked with repopulating. Right? So in our bone marrow, for example, right, we have our hematopoietic stem cells that can give rise to a dozen or so different types of blood cells. Right? But because that hematopoietic stem cell in our bone marrow is already far enough down this family tree of cell divisions, right? It's far enough down that it can only become whatever is sort of below it, like white blood cells, red blood cells, platelet forming cells, etc. And it cannot become something dramatically different like a nerve cell or a muscle cell, right? Because those would have branched off this family tree higher up. So the cells in our body today are mainly fully differentiated cells with a population of multipotent stem cells that persist indefinitely, right? And these fully differentiated cells, they will go on to proliferate only a few times more, right? Maybe 3, maybe 4 or 5 times before they undergo apoptosis and are replaced. Or quite frankly, so many of our fully differentiated cells are just physically lost, right? Because they line certain surfaces in the body that are subject to things like physical disruption and actually just being shed. These cells are shed from the epithelial layers, right? But the multipotent stem cells are sticking around and they are dividing to repopulate those. So from one single cell to 37 trillion cells, right? That's you today. And I think out of that 37 trillion cells or so, something crazy like, like 22 trillion of them are just red blood cells, right? And the remaining 11 trillion cells, only 11 trillion cells, right, are your nucleated cells. Right? Wouldn't it be cool if you could actually follow, right, that one cell from the start and actually map out over 20 something years, right, all the different divisions and commitments that were made so that you could make a humongous family tree? I mean, that would be pretty awesome, right? Because that literally could be then you could choose a cell in your body, like a skin cell, and trace it back and figure out along the way where it came from. So obviously we cannot do that in our bodies because there's far too many cells to do that. But there are a lot of organisms that are simpler than we are, right? And in fact, scientists have done this actual exercise using the cells of nematodes. So C. elegans is a very commonly studied model organism, right? For many reasons. They're easy to kind of manage in the lab. You can breed them, you can do mutations, things like that. But what's really interesting about it is an adult C. elegans has 959 somatic cells in its body, right? So that's kind of cool when you say, like, well, when you talk about a human, we use big sort of general numbers like, I don't know, 37 trillion or something, give or take, right? But here you have an organism that's small enough that you can actually say its genetic programming, right, dictates that it will have 959 somatic cells that make up its body. And that means you could feasibly try to map out this crazy family tree, starting from one fertilized egg, right, to an adult worm. And they've done that. So you might think like, well, that's really cutting edge stuff then if they were able to do that. No, actually, the reality is they did this several decades ago. Right. So this is the family tree. So starting from one single fertilized egg, right? The very first division, right? There's one line that goes over here, that's one daughter cell, and one line that goes over here, that's the other daughter cell, right? So scientists have been able to follow every division along the way to finally give rise to 959 cells that make up the body here. Right? And so then the tissues of the nervous system, reproductive system, the skin, or I don't even know if you call it a skin, I guess you call it a skin in the worm, and digestive tract, right? They're all mapped out. So the important thing, though, is even though you're dealing with a relatively small number of cells compared to something like us, right, all of these fully differentiated cells at the very bottom of each of these groups, right, they know when to stop. And that's the remarkable thing. So this whole idea of 959 cells, it's not 959 plus or minus 20 or 50, depending on how big the worm is. No, right? The worm is actually programmed in its genes to only have 959 cells. So as it loses some of those cells are replaced, right, in a very regulated manner. So even in a worm, proliferation of terminally differentiated cells is highly controlled and highly limited. We can see this in the lab, that fully differentiated cells are not designed to proliferate indefinitely. So fully differentiated cells, if you put them in a dish, in a tissue culture dish, and you give them some nutrients, media, oxygen, CO2, whatever, they will only divide a few times before they just stop. And when they stop, it doesn't mean that they die right away, right? They just senesce. So senesce means they take a break from the cell cycle. They exit. I said the cell cycle is like a freeway, right, going around, and G0 is like an exit ramp that you take, right? We said previously that cells can take a break and go into G0 if the environment is not right or conducive for them to be able to function properly in G1. But if things change, they could always get back onto the freeway, right? Get on the on-ramp. But senescence is different, right? Senescence is not the same as G0 arrest. It's a special type of G0 arrest, because when cells senesce, but they cannot go back and resume the cell cycle. So even when you have cell lines, and we'll talk about cell lines in a moment, cell lines that are typically these model cells that many research groups use, right, as sort of standard models of doing different types of cell experimentation. Cell lines are cells that can divide many, many more times than your typical natural differentiated cell. But even those cell lines, if you culture them and passage them too many times, right, they will senesce as well. So you look at this here. This is illustrating that on the x-axis is how many days in culture somebody in the lab is trying to maintain a population of cells, right? So as these cells grow and they divide, they fill up the dish, right? If they get too full, right, because then you have all kinds of like contact inhibition and things like that, right? To keep them happy and healthy, you might harvest those cells and divide them and put them into two new dishes so that they can keep growing, right? So each time you do that, we call that one passage, right? One generation to the next generation as you are passaging these cells in culture. So over the course of, let's say, 290, 300 days, you'd be expected to have to passage these cells numerous times, right? Depending upon how densely you are seeding the new dishes, splitting up the cells that are growing. On the y-axis is the how many doublings you'd get here. And you can see, right, that as you start off with, right, if you only have a few cells, for example, right, it takes them a little while to ramp up, right? Because if there's only a few cells, then yeah, they have plenty of space to divide, but maybe at first, they're trying to get acclimated to the media, to the culture, right? So the number of doublings starts off kind of slow, but at a certain point, so that's like your phase one, right? For example, if you just took these cells out of the freezer and you're trying to get them starting to grow again in the lab, right? At a certain point, they will happily hit a a accelerated growth phase. So that's phase 2. So this is now when they're really happy and they're doubling, and you're passaging them. But look, after a certain number of doublings, these cell line cells, right, after 40 or 50 doublings or so, right, they begin to taper off and they stop doubling. They stop moving through the cell cycle, and you reach phase 3, which is senescence. So once in senescence, cells cannot re-enter the replicative cell cycle. It doesn't mean they die, though, right? So they're in this kind of stasis. They remain viable, but importantly, they are phenotypically unresponsive anymore to extracellular signals like growth factors. And the reason is the signaling pathways inside of the cell, the mitogenic pathways, have essentially been turned off, right? So the cell is not dying, but it's just kind of sitting there. So you can see that here. So here are some, these are probably some sort of epithelial cells, or maybe they're fibroblasts. Normally, right, when they're happy and growing, they have this type of phenotype. But once they've hit senescence and been sort of encouraged to divide too many times beyond what they're programmed to do, they start taking on this funny shape. They kind of look like a fried egg, right? They're a lot more sort of spread out, and their nuclei, take on this kind of funny kind of staining. All right. So why is this important? Right? So normal differentiated cells in your body, not the fancy modified cell lines in the lab, like we were looking at the previous slide, because those cell lines you could passage 10, 20, 30, 40 times, and they'll just keep growing, even though they'll taper off maybe after 50 doublings, right? A typical fully differentiated cell in your body, like the keratinocytes that make your skin, right, that make your dermis and epidermis, they only proliferate maybe a handful of times. Like I said, maybe 3, maybe 5 times before they just stop, right? And then they are recycled by your body, or in the case of something like your skin, right, they end up just being lost physically. So we rely upon, in your skin, just like your other tissues of your body, there's going to be some population of multipotent stem cells mixed in here, right, that are constantly replenishing, right, through dividing and making more daughter cells, constantly replenishing the fully differentiated epithelial cells that are being lost. And that's how we're designed. So if we look at this here, here is a typical histology slide of healthy skin from an 18-year-old. So you can see here, the underlying pink area is the supporting tissue, maybe the stroma or connective tissue. The darker purple staining ones are the keratinocytes, right? So these are the fully differentiated cell types that are only going to divide a few more times before many of them just die or are recycled or lost, right? And then the keratin that they produce, the keratin protein, forms these nice, tough, dry keratin layers, which is our tough epidermal protection. So mixed in here, you cannot really tell, are some multipotent cells, right, that are dividing. So let's compare that to the skin of a 76-year-old. So the same cell types exist and are present, but you can see things look quite a bit different, right? You can tell, you can appreciate that there's less proliferative potential of these stem cells, right? So they are not as actively replenishing the keratinocytes. So you have fewer keratinocytes, right? And so therefore, the protective skin is a lot more frail. It's thinner in the elderly. We all know this, right? When we have older relatives, or we're just used to seeing, senior citizens who have skin that's just very, very sensitive and is, broken easily, right? Because you can look at the keratin layer, right? The quality of the keratin layer is also compromised. But you say, wait a minute. Why are there fewer keratinocytes if you still have stem cells down here? It's because over time, even the stem cells lose their proliferative abilities. Right? Why? Because remember the very first slide of this lecture when I showed you the thought question about photocopying photocopies, right? And every time you make a photocopy of a photocopy, you risk losing information, right? These multipotent stem cells that have persisted in your body for decades, or by the time you're, almost 80 years old, they've persisted for decades, right? They've been dividing, busy replenishing all of these keratinocytes through the many years of your life. But every time they divide, right, one of the daughter cells remains sort of as the stem cell, as the other daughter cell divides to replenish. You're simply going down that pathway of photocopies, of photocopies, of photocopies, right? So after several decades of life, you can imagine there will be a buildup, a natural buildup of mutations, not because of exposures to things necessarily, but just because of the loss of information coming from photocopying a photocopy of a photocopy, right? And that not only results in mutations that result in loss of proliferative potential, because after all, the ability of a cell to proliferate, right, is all driven by its genes and proteins, right? So compromises in the genetic sequence can interfere with the proliferative potential, but also think about this, right? Any mutations that are introduced along the way become part of that next set of photocopies, and any photocopies after that carry that baggage of that mutation with them. So the quality of the keratinocytes themselves, the fully differentiated cells, right, over time are getting lower and lower quality, right? So in contrast to this, consider that cancer cells, as well as those original embryonic stem cells, they have this feature of immortality about them, right? So let's discuss, what does it mean to be immortal? When discussing cancer cell lines, I think any good history lesson about cancer cell lines has to mention HeLa cells. I think several of you are already familiar with HeLa cells and the story of where HeLa cells came from, but it's nice to kind of revisit the story. So HeLa cells are the first established cancer cell line, and they were introduced into culture in early 1950s. And they came from a cervical cancer biopsy from this patient. This is Henrietta Lacks, who, she was being treated for cervical cancer. Her physician took some swabs of the cancer or the cervical epithelium, right? And her physician decided to try to introduce some of these cells and get them into culture, growing in culture. I mean, the thing that's amazing is these days, that type of thing would be completely unheard of because at the time, the doctor, didn't actually even ask permission, right, from Henrietta Lacks to have her cells be put into the lab and try to be grown, right? These days, that would be such an egregious violation of patient protection and HIPAA, right? But back then, sort of like anything goes, right? So some of these cells did grow in culture, right? Henrietta Lacks herself died later that year from her cancer, but her cells live on, right? And so these HeLa cells, which are named after her, to this day are used in labs all over the world. But what has happened is that with over time, many, many years of culturing these cells and passaging them and sharing them, right, more and more mutations have occurred randomly, right, in different HeLa cells, to the point where now there's no such thing as just one HeLa cell line, right? There are a whole number of different lines of HeLa cells. And here are 4 images of 4 different ones. All of 4 of these are technically HeLa cells, but you can tell they're very different in terms of their cell biology inside, okay? So to date, it's estimated that there's over 2 tons of HeLa cells being grown in labs right now, right? And the crazy thing about that is, you think about it, 2 tons of Henrietta Lacks cells, right? That's many times the total number of cells in Miss Lacks's own body. right, 70 years ago. And these cells have mutated so much, it's gotten to the point where the genomes inside of them are far enough from normal human genomes that there are actually some people who want to classify certain lines of HeLa cells as a brand new organism, right? It's kind of weird, right? I'm not sure I particularly subscribe to that, but that just gives you an idea, right? That these cells are immortal. They can keep growing and growing and growing. So in order for a cancer cell, remember, cancer starts from one specific cell that gets hit with, a mutation or a set of mutations that starts it down this pathway, right, of going rogue and transforming. One fundamental question to keep in mind is how long does it actually take for one transforming cell to eventually lead to the formation of a tumor, and how long does it take before the uncontrolled proliferation of those tumor cells results in a tumor that's just of a size of a mass that is lethal, right? So here, this data is interesting. If you look on the x-axis, this is tracking the number of tumor cell population doublings, right, and trying to correlate that with the diameter, the physical diameter of a resulting solid tumor. So how many cells correspond to a particular size tumor? You can see that when the cells have reached about 10 to the 8 cells, so that's about maybe something like 27 doublings of the population, that's about the size where the tumor is big enough. We're talking about maybe like 7 millimeters or so in diameter, where it can be first visible on an X-ray. Then by the time it reaches 10 to the 9 cells, that's about the size. So now it's about 10 millimeters, right? For certain types of cancers, that's the first time that the cancer is palpable, right? So for example, you imagine a lump in breast tissue. Things really take off after that, because by the time you get to 10 to the 12 cells, so then by then, right, the size of the diameter is about 100 millimeters. That is usually the size of a tumor that is considered, lethal. Right? So that happens after about 40 tumor cell population doublings. So does that surprise you, right? That it takes about 40 tumor cell population doublings in order to reach that number of tumor cells, right? That would give you a tumor mass that's big enough to be considered lethal, right? Let's investigate this. Do tumor cells really expand without restriction? If we think about one starting cell up here, generation 1, after about, well, and not after about, but after 7 generations, right, this would be 6 rounds of doubling, you'd go from 1 cell to 64 cells, right? So if you continue down this path, you would say, all right, if I went all the way to 40 generations, right? And let's say, for example, that if this is a cell that has lost control of proliferation, so that it is actually dividing, once a day, right? Let's say it divides once a day, right? So 40 generations, 40 days, right? You think, well, that doesn't seem right, right? To go from one transformed cell to a lethal tumor mass in 40 days. That doesn't make sense, right? Because the reality is a tumor takes many years to reach that critical mass of being fatal. So why would it take 5 to 10 years of a tumor growing if in reality it only takes 40 generations of dividing cells to reach that 10 to the 12 number of cells? It's because, think about this, right? Is 40 generations actually enough mitosis and enough opportunity for all the different mutations to build up that lead to something like an aggressive tumor? And hopefully you realize, no, that doesn't make sense, right? Because it's not like with every generation, you're going to get necessarily a new set of mutations that's then going to cause, the cells to become more aggressive and get passed on. And so the key here is tumor cells undergo substantial apoptosis, we've been learning about the importance of apoptosis, right? That if a cell detects that there's something wrong with itself, with its genome or whatever, right, that it cannot repair, it may halt the cell cycle with the help of p53, right? But if it cannot repair the problems, then the cell undergoes apoptosis, right? p53 helps to push the nuclear option, the button, and the cell undergoes apoptosis. So, not surprisingly, hopefully, right, if you imagine a transforming cell that first has a few mutations in it, as it divides and as the daughter cells encounter those types of problems within themselves that they cannot repair, the majority of these dividing cells will do the right thing and undergo apoptosis and take themselves out of the picture. In other words, this one starting cell that has the first hit or first mutation, right, as it divides, let's say this is a mutation that by chance is making its oncogenic pathways go out of control, right? Or it's a mutation that is somehow releasing a set of brakes. You can imagine that when it first divides, maybe one out of the two daughter cells, right, undergoes apoptosis properly, right? But maybe this other daughter cell, for whatever reason, sticks around. But when it divides, right, one of its daughter cells undergoes apoptosis, and so on and so forth. Right? So therefore, these transformed cells have to undergo many more than just 40 rounds of replication. In fact, if you look at this, if we say from generation 1 to generation 12 in this cartoon, right, if these cells were allowed to just keep dividing and dividing without any interference, without any apoptosis happening, then you'd predict that after 12 rounds, you'd already have 2 to the 11 cells. But instead, what do we have? We have at the bottom 1, 2, 3, 4, 5. Only 5 viable cells, right? So this is important to keep in mind because this is why cancer takes many years, right, to develop a sizable tumor mass. Now, of course, the actual time it takes is dependent upon the type of cancer you're talking about, right? And right, why that matters is because different types of cancers start from different normal cell types of origin, right? It all kind of goes back to this, what we were trying to appreciate since the start of the semester, right? Depending upon what type of cell starts the cancer, the intracellular pathways that are utilized by that particular type of cell, right, can have a big influence on how quickly or how slowly it moves through these generations of proliferation. Anyhow, so the bottom line is it takes many generations to reach a cell number in a detectable tumor mass, often many years. Right? And that's why many more generations means much more time, right? Much more time to get hit with additional mutations. Right? So not surprisingly, the progress of these cells dividing and proliferating is not linear, right? The rate at which these cancer cells divide to be the next generation and next generation is not a straight line. It is exponential because with additional random mutations being built up along the way or being accrued along the way and added to the existing mutations they already have, right? Certain mutations will bias those cells to be more aggressive and divide faster and faster and faster, right? So as you get towards the end, the growth of the tumor really starts to take off. That is why, right? You know this, that patients oftentimes, when they're first diagnosed with a tumor, it seems like, very sadly, that it's only a matter of months from the time that that tumor was first detected to when that tumor has reached a fatal mass, right? So you say to yourself, that's weird, because if what we're saying is it took several years for that tumor to get to that point that it was detectable, right? How come it only takes a few more months for it to get to a lethal tumor mass? It's because the rate of tumor growth increases exponentially as more and more mutations are piling up. So let's look at how certain factors like oncogenic proteins can prevent senescence from happening. Let's look at some human embryonic kidney cells. This is a popular cell line, these hex cells. So we'll represent them with this gray line on this figure here. So once again, we are following on the x-axis how many days in culture a lab technician is trying to grow and culture these hex cells. And then on the y-axis, again, is keeping track of the number of population doublings. The gray line is the normal cell line, all right, of these hex cells. So they have been transformed so that they do multiply more than a typical differentiated cell does. Right? But you can see, starting from 0 after about 10 days or so this would be about 5 passages or 5 doublings, the hex cells, the gray line, they plateau out, right? They don't die, but they hit senescence, right? So maybe for your experiments, if you're using the hex cells, you know that from the time you take the hex cells out of the freezer and you start growing them, right, you might want to use those hex cells within the first, 10 days or so, within the first 5 passages, in order to have the hex cells, phenotypically behaving normally for the sake of you doing experiments on them. But let's look, what happens if, as part of this experiment, you put into these cells different expression constructs like plasmids, DNA expression vectors that express 3 different versions of viral large T antigen. Right? So those would be represented by blue, green, or brown. Let's say the blue one is, these are the hex cells that have been transfected, right, or have wild type SV40 large T antigen put in them. We already learned that large T antigen, right, works by inhibiting the ability of retinoblastoma, or the RB gene, to function, right, to interact properly with things like E2Fs. We've also looked at how large T antigen is associated with P53. Remember, large T antigen, it forms this big star or snowflake-like structure and holds onto or sequesters P53. Right? So we can imagine that the wild type large T antigen in these hex cells is doing that. It's interfering with tumor suppressor genes like P53 and RB, and as a result, wow, what a profound effect, right? These hex cells that have large T antigen in them, they don't stop, right? They keep dividing and dividing and dividing with no senescence in sight, right? You can really appreciate that it really is this large T antigen doing it, because let's say the green experimental permutation is a construct of mutant large T antigen that lacks the domain that interacts with RB, right? So this mutant large T antigen cannot block the RB E2F associations, right? So as a result, you can see those cells also senesce more or less normally, right? And this brown line are manipulations of large T antigen that are mutated so that they cannot interact with P53, right? And you can see the brown line also, those cells behave more or less as expected, right? So that really highlights, right, that the ability of large T antigen to prevent senescence and allow cells to become immortal is a function of it needing to be able to interact with RB and P53. Okay. So normally, in a cell in your body that is dividing, a perfectly normal, healthy, differentiated cell, right? Once it's divided a few times and it needs to stop, right? As I said before, it doesn't necessarily mean that that cell dies, right? It doesn't necessarily mean it undergoes apoptosis. Right? It just kind of stops and eventually is going to be recycled or potentially physically lost. So how does that fully differentiated cell put the brakes on the cell cycle? Right? It does so by upregulating the brakes that it has at its disposal. Right? So certain brakes like the CDK inhibitors. Remember, these are things like P21, P16 that are important tumor suppressor genes because they associate with cyclin-dependent kinases and cyclins and prevent those cyclin CDKs from continuing to drive the cell cycle forward. So here you can see you have some cells here that you're growing. The top row of numbers is the number of population doublings of this particular cell line that you're growing. And what you are measuring here in a Western blot is levels of P21 and P16 proteins building up inside of these cells. You can see over time, even though these cells are dividing, right, to form daughter cells, if you were to measure how much P21 and P16 is in each next generation of these daughter cells, you find, right, that these daughter cells increasingly are filling up with these CDK inhibitors. So P21, P16, remember they work by inhibiting RB hyperphosphorylation because they are preventing the cyclin and CDKs from doing the hyperphosphorylation of RB. Here you can see morphologically, this is kind of neat. These are your active cells that are growing here. Right? And if you were to overexpress P16 in them experimentally, so you put in some sort of P16 expression construct so that these cells that were very active now are suddenly making a lot of P16 in them, you can look at the change in the morphology of these cells, right? They went from being the nicely formed, differentiated epithelial cells to this kind of fried egg sort of appearance, right? And if you look at the cytoskeleton, wow, right? You look at how dramatically the cytoskeleton has changed, right? It's filled with things like these microtubules now that basically cause the cell to kind of seize up, right? Because remember, the microtubules are like the network of different roads and things that things get transported around in there, right? Suddenly you have this big mess in there and the cell just no longer functions, right? It doesn't die, but it's just kind of sitting there until it can be recycled. Right? But that's in an experimentally overexpressed cell where P16 is made. But if you look at a naturally senesced cell due to just several cell divisions, you see the same thing, right? And so a cell that naturally has begun to upregulate its P16 after several divisions, you can appreciate, right? The same type of morphological changes are happening, and therefore this fully differentiated cell stops. Right? So whether we're talking about that experiment with the worm, the nematode, starting from one egg and following all 959 cells where they came from, and I said, even then, those fully differentiated cells at the bottom, right, know when to stop, right? Because when the time is right, after they divided a few more times, they become like this, right? They stop, right? And then they're just lost, either physically or they're recycled, and then you have stem cells replacing them, right? That's true of the worm. That's also true of our bodies, right? And that's why I said, quite literally, thank goodness the fully differentiated cells that make up me and make up you know when to stop, right? Because otherwise, right, I wouldn't look like this. I would just be one humongous, right, out of control tumor, right? And that's obviously not compatible with life. Right? All right, last slide. Here is some evidence of senescence in living tissues. So if we look at, for example, in vivo, right? So we have an example here of an adenoma forming. So a glandular tumor, it's not cancer yet necessarily, but this is happening in the gastrointestinal tract. So you can appreciate that if you stain these transforming cells with different protein markers that help identify which cells are proliferating and which cells are senescing, right? You can see something like this. Right? Certain cells still have the ability, right, to make P16 to put the brakes on. So the brown stain are the cells that are overexpressing P16 in them. Right? Contrast that to the ones that are stained brown in this slide. They are using an antibody not against the P16 protein, but against Ki67. So Ki67 is a very popular marker. It is a protein that's found in the nuclei of only actively dividing cells. So cells that are currently undergoing mitosis and proliferating express Ki67. So you can really see the contrast here, right? The cells that express high levels of P16, they're the ones that are not expressing high levels of Ki67 and vice versa, right? So I think this is a really nice way to kind of appreciate that the fully differentiated cells in our body, right, normally monitor themselves and prevent themselves from dividing too many times. So senescence is a very important part of normal cell behavior. Right? And so as we will study the next lecture, we'll take a closer look then at the changes that are taking place inside of cancer that bypasses this senescence. Right? We already looked at that data with the large T antigen, right? But obviously, right, there are many more things that are happening than just relying upon a viral protein to cause cancer. Okay. All right, let's go ahead and stop there.